DNA replication. First off, DNA is a nucleic acid made of nucleotides, and the parts of a nucleotide are the phosphate group, the sugar, which in this case is deoxyribose for DNA, which is why DNA is called deoxyribonucleic acid. But yeah, and the third part of a nucleotide is the nitrogenous base. DNA is made of many nucleotides. The bases are very important in DNA because the sequence of them determines an organism's traits. In DNA, the four bases are either adenine, thymine, cytosine, or guanine, and cytosine and guanine will always pair together, and adenine and thymine will always pair together. In DNA, the RNA, it's a little different, but that's not important right now. Just replace thymine for yours on RNA. Alright, back to DNA. DNA is basically two long strands of nucleotides put together by hydrogen bonds and then twirled, which makes a double helix shape. The sides of DNA are the phosphate groups, the deoxyribose sugar, and the middle part is bases. The two strands in DNA are anti-parallel, which means they are kind of put together. They're put together kind of like this, in an opposite direction. The role of DNA and the reason why it is so important is because it is your genetic code. DNA is used as the information needed to create things in your body. It can also give the instructions to make protein, which is needed for many things in the body, and it can determine what traits you might have, like hair color or eye color, and it is used to make cells. Cell differentiation is when your cells differentiate, obviously. But they differentiate by turning on some genes in your DNA and by turning off others. Your cells all use the same DNA, but they have different functions, like how your stomach cells have different functions than your eyeball cells. Cell differentiation is what gives them those different functions. Environmental factors can influence cell differentiation, like how temperature can influence a fox's fur coat to change to white in the winter, but when it turns back into summer, the fox's fur coat returns back to an orange color. Cell differentiation is cool and important, but it starts with stem cells, which involves DNA, which goes back to the point, DNA replication. In order to replicate DNA, we have to untwirl it, unzip it, and then with the help of enzymes, replicate it. The first step is separating the two strands of nucleotides, or unzipping them, I guess you could call it. This is done by an enzyme called helicase, and results in the formation of a replication fork. And it's not like the one you eat with. The replication fork has two strands, the leading strand and the lagging strand. For now, you'll just think of it as two strands. I'll explain that later. The two strands each provide a template for creating a new strand to DNA. Then the enzyme called primase comes down and places down primers, which is like a piece for DNA polymerase to start on and fill in the rest. The leading strand is the easiest to replicate because it works out nicely for DNA polymerase, because DNA polymerase can only add bases in one direction, from the 5' to the 3' end. And if you remember, I said that DNA is two strands of nucleotides put together, but they're anti-parallel, which means that if it's like this in that direction, it'll be like this and that in this direction, because that's what anti-parallel means. So because of this, we have to replicate the lagging strand differently in things called Okazaki fragments, which sounds Japanese and I like it. Alright, so Okazaki fragments work like this. So we have our lagging strand, and we place down primers every so often, and then your DNA polymerase goes from 5' to 3', fills it in, and then goes to the other primers from the 5' to 3' end and fills them in. Then, once the leading strand and the lagging strand are done, an enzyme called exonuclease removes the RNA primers from both strands, and then another DNA polymerase fills in the blanks where the RNA primers used to be in both strands. And then DNA ligase comes in and seals the fragments in both strands. And then hooray, once all that's done, we have duplicated DNA. The whole process is called semi-conservative because it takes two halves of your old DNA and creates two new halves. Combine each old half with a new half, and then you have two genetically identical pieces of DNA. DNA's components are common in all organisms, even in single-celled prokaryotes, because they all contain DNA, genetic code, which is important for them to function properly. And whenever they reproduce, they replicate their DNA, which is a process called DNA replication. And thanks for watching. Alright.